poster titled Role of Receptor Protein Tyrosine Phosphatases and Their Relationship with Coenzyme Sulfate Proteoglycans During Spinal Cord Injury, and my target audience will be experts within the field of nerve regeneration. As part of the introduction, I would like to go over the basic elements of spinal cord injury. Looking at the figure on the left, we can see that injury to the spinal cord will result in damaged axons that are either crushed or completely severed, and the extent of the damage done to the axons depends on the injury severity. Transitioning to the figure on the right, we can see that following the injury, axons remain demyelinated and severed, and distal segments of the severed axon will undergo Wallerian degeneration as the proximal stump attempts to regenerate and innervate distal targets, but is unsuccessful because of the glial scar. The injury induces an immune-associated microglial response that promotes and activates astrocytes, glial precursors, microglia, and fibroblasts to form a glial scar around the lesion site. And the astrocytes that make up that glial scar serve as a physical barrier that impedes axonal regeneration. And the glial scar also upregulates growth inhibitory molecules to further inhibit axonal outgrowth, in addition to establishing a physical barrier. And one of the many molecules that are upregulated that impedes axonal regeneration are CSPGs, which are considered within this study. One of the many receptors that interacts with the inhibitory molecules secreted by the glial scar are receptor tyrosine phosphatases, also known as RPTPs. RPTPs are important mediators of signal transduction of the plasma membrane and play a role in many events, including cell growth and proliferation and neural development. The figure shown in front of you presents many different types of receptor tyrosine phosphatases and are classified based on whether there is a single or tandem of highly conserved intracellular phosphotyrosine-specific phosphatase domains. But the specific receptor that will be considered and analyzed in the study is RPTP sigma, circled in red, and is a member of the type 2A family, circled in blue. So the figure shown is a schematic representation of how the glial scar interacts and impedes axonal outgrowth. So looking at this figure, we can see the glial scar is composed of reactive astrocytes shown in blue at the very top of the figure, and these reactive astrocytes will secrete many different types of inhibitory molecules such as CSPGs, NOGO, OMGP, RGMA, etc. But the specific inhibitory molecule of interest in this study is CSPGs and their interaction with PTP sigma, although they interact with another receptor also known as LAR. But when CSPGs, shown in the very left, interact and bind to PTP sigma, that will thereby activate the receptor, which then induces signal changes in the ROROC signal cascade, which is a downstream signaling pathway within the neuron. An activation of this signaling cascade within the neuron then results in growth cone collapse, neurite retraction, and apoptosis, thereby impeding axonal regeneration and preventing successful reinnervation of the distal targets within the central nervous system. Goals of the study consist of 4 elements. One is deriving a structure for the RPTP sigma receptor. Second is analyzing the structural features and properties of this receptor. 3 is understanding the binding interaction between the receptor and CSPGs. And 4 is deriving a method to disrupt the interaction between the receptor and inhibitory molecule to therefore improve axonal regeneration and functional behavioral recovery after injury. So here's a brief description of the methods that were used in the study. First, the receptor was filtered via nickel affinity chromatography and then was crystallized in sitting drop vapor diffusion format. After the receptor was crystallized, X-ray diffraction data was collected from those crystals to determine the receptor structure. And the wedge peptide that was synthesized using GenScript and assessed using mass spectrometry was used to disrupt the interaction between the receptor and inhibitory molecule to improve regeneration. So the first experimental results obtained within the study concerned the domain organization and structure of type 2A RPTP sigma. Figure A provides a schematic representation of the domain organization of the receptor and also provides an insight into the common domain architecture that is present within the type 2A family. Looking at figure A, we can see that the receptor is composed of an extracellular region that contains 3 Ig domains and 9 Fn repeats, and then a single transmembrane helix followed by a tandem intracellular phosphatase domain that is monomeric in solution. And the variation of type 2A RPTPs is generated via alternative splicing, specifically within the ectodomain. Figure B below Figure A provides a cartoon illustration of the Ig and Fn domains of the receptor and is specifically color-coded in accordance with Figure A. And Figure B demonstrates the flexibility of the ectodomain and the general conformation of the Ig and Fn domains. The second experimental result obtained in the study consisted of the structure and general conformation of the Ig1 and Ig2 domains. So figure A on the left shows the general conformation of Ig1 and Ig2, with Ig1 being shown in light tan and Ig2 being shown in dark blue. But specifically, figure A highlights the linker region shown in the black box and the domain interface shown in the red box. Transitioning the figure B on the right, we can see that figure B represents the linker region that was highlighted with the black box in figure A. And we can see that figure B is made up of hydrophobic residues that include P130, L124, V44, etc. And the side chains of residues P130, L124, L129, and F133 pack very closely with I42, V44, and A212 to form a hydrophobic interdomain region. Lastly, figure C presents the interactions observed at the domain interface between Ig1 and Ig2, which was highlighted in figure A in the previous slide as the red box. And what figure C shows is that there are many interactions between residues of Ig1 and Ig2, which include salt bridges and hydrogen bonds. And the salt bridges are represented as the black dashed lines and include residues R91, E205, E215, and R215. But in addition to the salt bridges, there are many hydrogen bonds present and shown in the green dashed lines and involve the side chains side chain amide of Q41 hydroxyl groups of S50 and Y216, backbone carbonyls of I42 and F171, backbone amide of V214, and the two water molecules that are shown in magenta. And these salt bridges and hydrogen bonds that were observed at this domain interface help in holding Ig1 and Ig2 in a rigid conformation. Another set of results obtained within the study concerned the electrostatic interaction observed between CSPG and RPTP sigma. So looking at the figure on the right, we can see the structure of Ig1 shown in tan, and more importantly, the residues that are important for binding CSPG shown as red sticks. And the residues that are important for coordinating the binding of CSPG to the receptor include K68, K67, K70, K71, K99, and K96. And these residues are all lysine residues, which is important to denote when we discuss figure B on the right, which is the solvent accessible surface of Ig1 and Ig2 domains shown as an electrostatic surface potential. And we can see that all the residues that are shown in figure A as red sticks that are important for binding are labeled and concentrated in the positively charged regions in figure B, which is expected considering that all the binding residues are lysine residues. Understanding which residues hold importance for binding and where their location is with regards to the electrostatic surface potential of the receptor, as shown in the previous slide, allowed for researchers to develop a model for binding of CSPGs to our PTP sigma as shown in the figure presented to you. And as said on the previous slide, the residues on the receptor that are important for CSPG binding are positively charged. The highly sulfated proteoglycan GAG chains, shown and denoted as CS in the figure, carry a strong negative charge and therefore will electrostatically interact with the positively charged residues on the receptor. So essentially, the binding of CSPG, which is negatively charged, binds to the positively charged residues on the receptor, and this binding will subsequently disrupt the clustering of the receptor and thereby fail to induce oligomerization. And failure to cluster does not create an uneven distribution of tyrosine phosphatase activity that can lead to microdomains with high phosphotyrosine levels that will support neural outgrowth. And as a result, axonal outgrowth is inhibited. The last set of results obtained within the study was with regards to developing a method to disrupt the interaction between the receptor and inhibitory molecule in an attempt to improve axonal outgrowth. So looking at figure A is the general structure of the receptor, and analyzing this structure led researchers to identify and discover a highly conserved 24 amino acid intracellular wedge domain, which is shown in red. And why this was interesting to researchers was because, as stated earlier in the introduction, when CSPGs bind to the receptor, that activates the receptor and results in a signal change in the downstream signaling pathway, specifically the ROCK signaling pathway that will then collapse growth cones and inhibit regeneration. And identifying this wedge domain was important because wedge domains are known to regulate those downstream signaling pathways, which was why researchers designed a intracellular sigma peptide, which is shown in figure B. And in figure B, we can see that the intracellular sigma peptide designed is a novel peptide mimetic of the PTP sigma wedge with a TAT domain to facilitate membrane penetration. Figure C at the very bottom is a time-lapse imaging of an adult sensory neuron growth cone after treatment with the sigma peptide. And the timestamps next to each sensory neuron indicates time in minutes. And we can see that sigma peptide treatment allowed adult sensory neurons to extend axons through a CSPG gradient in a dose-dependent manner. Although growth cones treated with the sigma peptide are still transiently collapsed by CSPGs, they continued to reform growth cones, which allowed them to eventually cross this inhibitory gradient made up of CSPGs. And sigma peptide treatment was sufficient to convert stabilized dystrophic growth cones into a motile state. So altogether, designing this intracellular sigma peptide to disrupt the interaction between the receptor inhibitory molecule allowed sensory neurons to reform growth cones and cross this inhibitory gradient, which will allow them to reinnervate distal targets and have successful regeneration and functional recovery of the axon. So the takeaways of the study was that the domain organization of the receptor includes Ig-like domains, FN domains, and phosphatase domains. And the linker region between the Ig1 and Ig2 domains is composed of residues that form a hydrophobic interdomain region, and the interaction at the domain interface between the Ig1 and Ig2 domains consists of salt bridges and a network of hydrogen bonds that contribute to the rigid conformation of the Ig-like domains. More importantly, the residues that are important for CSPG bindings are concentrated in a positively charged region, whereas the proteoglycan GAG chains carry a strong negative charge, thereby suggesting an electrostatic interaction between the receptor and inhibitory molecule. And the binding of CSPG to the receptor disrupts the clustering of our PTP sigma and fails to create an uneven distribution of tyrosine phosphatase activity that would support neural outgrowth. And by creating a peptide sequence with a TAT domain, disrupted the interaction between RPTP sigma and CSPGs to promote axonal outgrowth. The peptide converted dystrophic growth cones into a motile state that allowed axons to cross a inhibitory gradient made up of CSPGs and therefore regenerate and establish behavioral recovery.